Hey guys, this is the last part of macromolecules, and today we're going to finish proteins and nucleic acids. This is Dr. Yu. Welcome back to another lesson. All right, now let's look at proteins. Proteins are a super important group of micromolecules in our body. You probably remember that we talked about the DNA, as the blueprint for our body, and the DNA code for proteins, right? So eventually the genetic information in the blueprint is used to make I like to call proteins workers, the workers in our body, because proteins do pretty much everything. Right? They can be the structural component, they can be the enzymes that catalyze chemical reactions. They really perform a wide range of functions in our body. So proteins are polymers, and the monomers are amino acids. So when you look at a protein, it's really a chain. It does have that 3D kind of shape, three-dimensional shape. But once you kind of straighten it out, it's really kind of like a chain. So proteins are considered polymers, and each of these subunits, or the building block, so all these are amino acid. So AA stands for amino acid. So you do have to remember that the monomers for proteins are amino acids. You may see some questions on that on TEAS. Now, these amino acids are linked by peptide bond. So the bond over here, these are peptide bond. There you go. So that's the bond that linked all the amino acids together to form a chain, right? A polypeptide chain. There are 20 different amino acids, and all the proteins are composed of those 20 different amino acids. But of course, the order of amino acids in the chain is going to be different, right? And that kind of determines what shape molecule the protein molecule is going to have and what function the protein molecule is going to perform. All right, now let's look at the structure of amino acid. This is very important. It's mentioned in the TEAS study manual. So when you first look at the structure, you may think, oh, that's too complicated. I'm not good at chemistry. But we can really kind of break it down to different components. And once we do that, it's a lot easier to remember. Now, start with the central carbon. So you have a central carbon atom in the middle. And then we're going to build the other components around this central carbon. It's also known as the alpha carbon. Okay, so carbon can have 4 covalent bonds, right? And that's because carbon has 4 electrons in the valence shell. So it's kind of a number in between 1 and 8, right? It's kind of in the middle. So 4 is right here. So carbon atom does not like to lose all the 4 electrons or gain another 4 electrons to get to 8. So carbon usually shares these 4 electrons with other atoms to form covalent bond. So carbon usually can form up to 4 covalent bonds. All right, now we're going to add 4 components around this central carbon. All these 4 components form a covalent bond right here with carbon. Okay, so you can see that's 1, 2, 3, 4. Right, so let's add the 4 components. The first component is very simple. It's just a hydrogen atom. So that's number 1. That's easy enough, right? Okay. The second component is an amino group. Number 2 right here, amino group. So amino group is basically NH2. So you can see the amino group has a nitrogen atom. And then the nitrogen is going to form a bond with that carbon. Okay, so that's number 2 amino group. Number 3 is this carboxyl group. So that's number 3. Carboxyl group has a carbon double bonded with an oxygen right here. And then this carbon has another covalent bond with a hydroxyl group right here, OH. So if you need to write it out, that's going to be C carbon OOH. Right? So the O is this first O, oxygen, is the oxygen that has a double bond with carbon, and then the OH is the hydroxyl group. So that's the chemical formula for the hydroxyl group. And the very, very last component bonded with that central carbon is an R group, the R side chain. Now, the R can be anything. It can be as simple as a hydrogen atom. It could be as complex as a few chemical functional groups all linked together. Okay, so that's R. We talk about there are 20 different amino acids. The 20 amino acids only differ in the side chain. Okay, that's the only difference between those 20 different amino acids. Everything else is the same. Everybody has a central carbon, and that central carbon has a covalent bond with 3 components, right? Let me use a different color. Everybody has this hydrogen bonded to central carbon. Everybody has this amino group and this carbox group. Okay, so the only difference between the 20 different amino acids is this R side chain. Now, this variable R side chain also determines the chemical property for the amino acid. We talk about hydrophobic, and the opposite is hydrophilic. Okay. Hydro, that means water. Phobic means fear, right? Fear. If a chemical is hydrophobic, that means that chemical is not water soluble because the chemical fears water, right? And that's usually because the chemical is non-polar, right? Non-polar, so it does not mix well with water. It's not water soluble. So that's hydrophobic. Hydrophilic is the opposite. Philic, that means like, right? If a chemical is a hydrophilic, that means that chemical is normally water soluble. It likes water. It mixes with the water very well. Now, I'll give you some examples. Hydrophobic substances that we have gone over are lipids, right? Remember, all the lipids are water insoluble. They do not mix with the water. They do not like to interact with the water. So if you put fat or oil or that wax layer or steroids, if you put all those things in water, they are not going to mix with the water. All right. Now, some examples of hydrophilic substances: sodium chloride, calcium chloride, potassium chloride, and calcium chloride. So you probably notice all these things can dissolve in water very easily, right? When you cook, you put salt in your food, it dissolves pretty fast. Sugar, glucose or sucrose, the sugar that you usually have in your pantry, they also dissolve in water very easily. So the salts, the sugars, most of the smaller carbohydrates, disaccharide, monosaccharides, those simple sugars are usually very hydrophilic. Hey guys, I'm using this kind of special screen because I seem to have a lot of trouble with my pen. For some reason, it just doesn't draw anything. So I'm trying this new kind of layout. So hopefully it works. Now let's look at the variety of functions that proteins perform. So we're going to touch on the functions mentioned in the TEAS review, such as the enzyme, defense, transport. First, let's look at fibrous proteins. So these proteins are usually big protein fibers, and they're often hydrophobic. And the hydrophobic property really comes from the R groups or the side chains from the amino acids that make up these proteins, right? Good examples are keratin, right? You probably remember the protective protein fibers in nails, hair, and the superficial layers of skin cells. Also collagen, which can also be found in the skin, which gives you that kind of a bounciness of your skin and makes your skin plump. It looks young. Also, collagen is found in the bones. Remember, when we talk about bones, we talk about the inorganic component, which are mostly calcium phosphate, right? They give the bone that kind of tough texture, that hardness. So the hardness of the bone comes from the inorganic components. We also have organic components in the bone, such as collagen, right? Collagen makes the bone flexible, right? It gives the bone that flexible texture. Otherwise, the bones are going to be very brittle, right? Very kind of crispy, very easy to break. So the organic components, such as collagen, are also very important for bone health. Globular proteins are usually kind of have this globular kind of round shape. And usually these proteins have hydrophilic surface, meaning that these proteins are usually water soluble because their hydrophilic components, hydrophilic amino acids, are on the surface. So this makes these proteins water soluble. For instance, we have antibodies, right? Antibodies are a type of globular protein. And if you remember, when we talk about the immune system, we mentioned that the antibodies are part of the humoral immune response, right? And specifically, antibodies are produced by B cells. B cells. And if you want to be even more specific, it's a special group of a B cell called the plasma cells that produce antibodies, right? So that's pretty important information. Enzymes, the biological catalysts, right, are also proteins. Now, I just want to point out most of the enzymes are proteins, but there are some enzymes, a very small portion though, are RNAs. RNAs, we'll talk about RNAs in the nucleic acid chapter. So RNAs are a type of nucleic acids. They're similar to DNAs, but they're not exactly the same. So a small fraction of the enzymes are RNAs, but the majority of the enzymes are proteins. And the TEAS study manual does consider all the enzymes to be proteins. So if you see questions on TEAS about enzymes, just assume that they're asking you about the protein enzymes and not the RNA enzymes. The last example of globular protein is hemoglobin. Again, this is a super important protein, right? If you remember, hemoglobin is present inside red blood cells, right? Hemoglobins combine to oxygen and the carbon dioxide, which allows the red blood cells to transport these gas molecules throughout the body, right? So hemoglobin, very important. And in hemoglobin, there's iron, right? Fe, so that's the symbol for iron. If you are deficient in iron, then that's going to kind of disrupt the production of hemoglobin, right? So this may lead to anemia. All right, there are also some proteins that can be found in the cell membrane, and usually they have a sandwiched structure. So what does it mean? Now, let me draw the basic structure of the cell membrane. Earlier, when we talked about the lipids, we mentioned that the phospholipid is the backbone of the cell membrane. And if you remember probably a little bit that the phospholipid has a couple important components, right? There's this head. So the head is mainly the phosphate group. Phosphate group carries a negative charge, which makes the phosphate group polar or hydrophilic. So the phosphate head, so that's the head, likes water. All right. And connected to the phosphate head is two fatty acid chains. Right? So we'll call them the tail. So this is the fatty acid tails. Fatty acids are a group of lipids, right? So they do not like water. All lipids are not water soluble. All lipids are hydrophobic. So this kind of special characteristic really kind of determines the structure of phospholipid as the backbone of the cell membrane. If I have to draw it, it looks something like this. So the phosphate head, the polar head, is going to face outward because the phosphate heads like water, right? And just think of the cell membranes are usually in this watery environment, right? So the phosphate head is going to face outward, and the fatty acid tails are going to be inside. It's going to be face inward. And then you're going to have 2 layers of this phospholipid. And that's why we say the cell membrane backbone is a typical phospholipid bilayer, right? That bi means 2. So you have 2 layers of phospholipid. So this allows the fatty acid portion to be inside and not come into contact with water. So only the phosphate head interact with the water because they're on the exterior. All right, now I'm going to draw a little bit more over here, and then I will draw the proteins that are embedded in the cell membrane. Okay, all right. Now, usually these cell membrane proteins are embedded. Let me use a different color. Are embedded in the cell membrane. So you can see they kind of span through the entire length of the cell membrane, right? Okay. Now, the sandwich structure really just means that I'm going to use a different color. The surface of the embedded proteins, like over here, that portion and this portion, this is going to be hydrophilic, because they interacted with the water, right? If you think about it, the cell membrane is in this kind of watery environment, right? So on both sides, that's going to be mostly water. So the end that comes into contact with the water is going to be hydrophilic. And then the portion that's inside the cell membrane is going to be hydrophobic. I'm going to use the purple color. So this portion is going to be hydrophobic. So this portion is touching or is interacting with the fatty acid tails, right? So this portion has to be hydrophobic. Otherwise, the fatty acid tails would repel this portion of the protein. These cell membrane proteins perform, again, many functions. For example, they transport. Right? We mentioned earlier that the cell membrane has selective permeability. Selective permeability, meaning the cell membrane is permeable only to certain substances, but not others. And that's because the majority of the cell membrane is this fatty acid tail portion, right? The fatty acid tails only like hydrophobic molecules. So only molecules that are hydrophobic can cross the cell membrane freely. Hydrophilic substances will not be let in because the hydrophilic molecules are repelled by the fatty acid tails. So I'm going to just use these terms here. Hydrophilic, they cannot cross the cell membrane freely. Again, because they're repelled by the fatty acid tails. Repelled by fatty acid tails. Now, if it's a hydrophobic molecule, that's a totally different story, right? Usually they can cross freely because they mix well with the fatty acid tails, right? So they can usually cross the cell membrane very easily. Now, of course, the size of the molecule also affects whether the molecule can cross the membrane or not. But for now, we just focused on whether the molecule is hydrophobic or hydrophilic. But again, if you are taking a biology class, you have to consider the size of the molecule as well. Cell signaling. Some of these embedded proteins are used as receptors, which are involved in the cell signaling process. I don't know if you guys remember when we talk about the synapse, right? How a motor neuron controls a muscle movement. Remember the presynaptic, I'm drawing up here. This is the presynaptic portion of the synapse, right? So this could be an axon terminal that controls this particular muscle cell. This axon terminal can release chemical messengers, right? Which is acetylcholine. So acetylcholine is the chemical messenger that can diffuse through this synapse cleft and get to the muscle cells, and they're going to tell the muscle cells to contract, right? Okay, now on the surface of the muscle cell, there are receptors. Now, let me use a different color. So these are the receptors. And eventually, the acetylcholine will bind to the receptors, and this triggers a cascade of chemical reactions, which will eventually generate muscle contraction, right? So these green color receptors that bind to acetylcholine, those receptors are cell membrane proteins, right? So you can see that they are involved in the cell signaling process. Cell recognition on the cell surface, basically on the cell membrane, there are substances that have the protein components, and usually they're used as cell markers. For example, glycoproteins. Glyco refers to the sugar component. So this is a sugar protein complex. And some of the glycoproteins on the cell surface are used as cell markers. So they are involved in cell recognition. Okay, next, enzymes. Enzymes are a very important group of proteins. And if you look at the TEAS study manual, there is a question on enzymes. It tests your knowledge about what kind of molecules enzymes are. If enzymes are broken down, you will get the associated monomers that make up those enzyme molecules. And those monomers should be amino acids, right? Enzymes catalyze or speed up chemical reactions. Without enzymes in our body, the chemical reactions that are going on, the hundreds and thousands of chemical reactions, would take a very long time to proceed. So that's not realistic, right? So thanks to the enzymes, we can perform our normal body functions really well because we can complete those chemical reactions in milliseconds or seconds. So how do enzymes work? Basically, they lower the amount of energy required to start the reaction. Okay, so whenever a reaction is going to happen, usually it requires a little bit kind of startup energy to get the chemical reaction going. That amount of the energy kind of limits how fast the chemical reaction can go. But with the enzymes, that amount of startup energy is dramatically lowered. So you only need a little bit to get the chemical reaction going. So that's why enzymes can really kind of catalyze and speed up chemical reactions. Another key information about enzymes is the active site. If you look at the diagram down here, you will notice that this particular site, which looks like a notch, right? That's the active site. That's where the substrate binds to the enzyme, and this is also where the enzyme can kind of change the shape slightly and reduce the amount of energy needed and get the chemical reaction going like really rapidly. So that's the active site. Many factors can affect enzyme activity and the rate of chemical reaction. For example, temperature. Right? So usually when temperature is higher, enzyme activity increases. And that's because a higher temperature, higher energy for the molecules to be active, to collide, make the chemical reaction happen, right? So temperature usually has a positive impact on the enzyme. However, if the temperature is too high, it could denature the enzyme. Let me draw this graph to kind of show you the relationship between temperature and enzyme activity. When temperature, so that's temperature, and this is the enzyme activity. Right. When temperature goes up, the enzyme activity will also go up, right? So you can see temperature goes up from the left to right. The enzyme activity also increases, right? From bottom up. Okay. So that's the relationship. If the temperature reaches the optimal temperature for a specific enzyme, the enzyme is going to have this highest activity, right? So you can see this is the peak, right? That's the highest activity. If the temperature passes that point, so temperature keeps going up, right, keeps increasing, then enzyme activity is going to go down, it's going to decrease. And that's because higher temperature may denature the enzyme. If the temperature is too high, it's going to, destroy the biological molecules, right? So it has this kind of bell curve normally. So again, if the temperature within the optimal range goes up, enzyme activity goes up. But once you reach the optimal temperature, as the temperature increases, the enzyme activity is going to go down, right? Because it just gets, think about this, it just gets too hot for the enzyme. pH also affects enzyme activity. Usually, specific enzymes will have their optimal pH. So similar to this temperature graph, right? If the pH is at the optimal pH for the enzyme, then enzyme activity is going to be the highest, right? So on both sides of the optimal pH, the enzyme activity will decrease. Salts, that's another factor that could affect enzyme activity because salts have those ions, right? And those ions could disrupt the ionic bond within a protein structure between those amino acids. We sometimes use salts to preserve food, right? That's because salts can disrupt the bonds in proteins of microorganisms. So this can inhibit bacterial growth, which allows food to last longer.